So it may be very conventional and not so related to the topic, but on the other hand, we have discussed a lot here in the group between different people, also the impact of the food chain, and we think that the animal, the living animal, and the feed of the living animal is an important part of the chain. And you know that in different European projects, a lot of research has been done on feed additives, which is a highly important topic, so I think no need to go into details, but trace elements, phytogenic additives, probiotics, enzymes, and so on have been quite extensively studied. So we were considering a bit the role of zinc in pigs, which was a national project, a cooperative project, and we were quite interested to identify some mechanisms on how zinc would affect animal health and also the gut colonization with microbes. And you know that zinc is one of these elements which are really essential for almost everything. So we know more than 300 biological processes which are linked to an adequate zinc intake. And we know that it has a lot of functions in not only physiology but also in the regulation. So many aspects of gene regulation are also linked with zinc finger proteins, for instance. So interestingly, most of the work which has been done up to date was linked with zinc oxide, which is a very conventional form of zinc, which can be used for humans and also for animals. However, we must say that zinc oxide is already a topic in its own because it has a very different bioavailability. Nowadays we see that the industry provides farmers with many different forms of zinc oxide with different claims. Some of them are claiming that the availability is somehow lower, so you should have more zinc in the gut, which may have a deeper impact on the gut microbes. Others claim higher bioavailability, allowing farmers to use lower levels of trace elements in the feed, and by this supporting animal health. So it's a very diverse field in the moment, I would say. Besides that, we have special developments now with this organic bound zinc, so for instance bound to amino acids, which should have a higher bioavailability, but not much work has been actually done on that in terms of animal health and also gut function. So I will focus more or less here on this point on zinc oxide. And one important question is, what does it mean for the intestinal tract? I just want to show you some pictures from the intestinal development in piglets. You see 4 different piglets, and maybe we can vote. What do you think, which one is the healthy one? A, B, C, or D? What would you say? Okay, you're right, all are healthy. That means finally we have a high diversity already in the morphology of the gut, and we know that this morphology also changes, also the function changes based on different ages, diets, environment, health status, and so on. So we have a lot of factors which can be influenced, and we were quite interested to study a bit more this trace element host interaction in that context. So here you see one picture showing the small intestine, a villus, a cross-section of a villus with a mucus-secreting goblet cell. Here you see the same from the large intestine with a different staining. You see this mucus layer, which is considered as being protective. And we found out that zinc in the diet has a clear impact. Low zinc intakes, intermediate zinc intakes, and very high zinc intakes cause significant changes in the composition of the mucus which is produced in the gut. So, for those who are not so familiar with the story, zinc oxide is very often used in Europe as a feed additive to protect animal health in high dosages. So, the normal requirement would be covered by much lower doses compared to the so-called pharmacological levels. So, this one was a pharmacological level here, 2,500 milligram of zinc in the diet. This one was quite low, 60 milligram, and this was intermediate 150, which is somehow the legal maximum here in Europe. But in general, we have a practice that many farmers use much higher levels than are currently allowed, which means it induces an accumulation of zinc in the GI tract and for sure also for the environment. Here we did one study with, I would say, normal levels, 120 milligram, 200 and 3000 milligram of zinc from zinc oxide. And you see the concentrations in the different sections of the GI tract, which is here the colon or here the cecum. And the concentrations goes up to more than 12 gram per kilo diameter, which is really high. And this is meaning somehow that we have an environmental issue here because these high zinc levels are excreted, and we know in many regions in Europe we have these problems by zinc accumulation in the soil, which may mean also a problem for plant breedings, for plant growth. So this is one reason why we should be careful with using such an additive in high dosages amongst the legal issue. Here you see it's a very quick process. We used here this group with a high zinc level in the diet, 3000 ppm, and you see these were piglets after weaning. After 3 days, somehow it starts to rise in the feces compared to the other levels here, which were quite normal, so you get much lower levels. So that means clearly we have an environmental impact. But our question was, is the pig, is the animal, the organism as such protected against these high levels? Because it might induce something like a zinc toxicity. And to study that, we looked at data for the retention of zinc in the pig, but we also studied different transport proteins in the gut. So based on literature, we know in normal conditions the animal transports zinc by 2 transport proteins into the epithelial cell of the gut. There's another transport protein transporting zinc into the body. If there is a deficiency, short-term or long-term, there's an upregulation of these zinc transporters, and so by this there is a certain flexibility, and the pig can adopt as all other living animals or beings on this different zinc supply. But what happens when we have a high zinc supply? So our question was simply, would animals or this pig as a model be able to switch down these transport processes when it is exposed to high zinc levels? And indeed, that happens. So we have a downregulation of this LIP4 zinc transporter. We have an upregulation of a storage protein here, which allows to get rid of zinc by the normal cell sloughing. But we also see that these pigs upregulate this zinc transporter at the basolateral side, which means they put a lot of zinc into the body pool, which means finally we have an accumulation also in some organs like in the liver, in the kidney, but also in the bone. So finally, obviously, we get a lot of zinc into the body, which may be a toxic phenomenon finally. We know when you apply these high levels for more than 2 or 3 weeks, you get really also a problem with animal health and performance. So insofar, using these high zinc levels have a positive impact on animal health on the one side, so it reduces the risk for diarrhea. On the other hand, it can cause, after long-term application, also toxicity. But our main interest was really related to the intestinal microbiota, because this might give us some ideas about the mode of action of this trace element, also in terms of the health protective effect. And our research was directed towards this gut-associated microbes, so especially also the microbes associated in close relation here with the gut tissue. And interestingly, the intestinal microbiota has a certain zinc requirement, which is more or less known for most of the bacterial species. So we also found that depending on the dietary level of zinc in the diet, you get a different reaction of the intestinal microbes in pigs. So we know when the microbes have a deficiency of zinc, they have an impaired growth or metabolic function. When you have too much zinc in the environment, it can be toxic also to microbes. And it's very interesting that the gut microbes adapt to this situation. So we made a quite simple experiment. So we had piglets being fed normal zinc levels and pigs being fed high zinc levels. And we took these microbes from these piglets, took them to the lab and grew them under the condition of no zinc in the growth medium. You see the low zinc group and the high zinc group bacteria, similar growth rate. But when we added zinc, so made something like a zinc stress, the situation changes. So those bacteria coming from pigs being fed, so high zinc levels, have a better adaptation, so they grow faster compared to the bacteria coming from the normal zinc piglets. That means, obviously, the microbiota reacts to the trace element intake, which is not so surprising, but at least it shows there's something going on. And based on these findings, members of our group did some typing, so looked at different strains, reference strains, exposed them to different levels of zinc oxide, and to make it short, what we found out in that was that we actually can type intestinal bacteria based on their sensitivity to a trace element. So they can be susceptible, so growth inhibition happens very easily. They can be resistant, so no impairment of growth, and they can have something like an intermediate sensitivity. Why is it interesting? Because we and also others found that especially those gut bacteria that we call beneficial, so like lactobacilli and others, seem to be extremely sensitive to this trace element, while the enterobacteria here, for instance E. coli, seem to be very resistant. And I will show you some data on that. So, first of all, one interesting point is microbial diversity. This is an older study which was published in the 90s. And what they did at that time was to study the bacterial diversity in piglets after weaning. So weaning took place somehow here in this red area. And you see the diversity which was measured at that time based on phenotypic data was decreased, the diversity was decreased in the control pigs, and it was maintained somehow higher in the piglets fed high levels of zinc oxide. So obviously this stress which occurs during weaning period was alleviated by the high levels of zinc oxide. We have seen similar effects of these high levels of zinc on the gut microbiota. So what we did actually was to make something like a fingerprint in a